Hi to all of you. In this particular session, we'll try to revise and recap just the carbohydrate metabolism, probably in 15 minutes, nothing more than that. Okay, so this is not an attempt to teach you biochemistry, this is only an attempt to just recap, maybe let's say whatever you have studied about carbohydrate metabolism in a quick 10 to 15 minutes review. Okay, so before we start, I know that your NEET exams are around the corner, and as it is said, you can't control everything. Sometimes you just need to relax and have faith that things will work out. Let go a little and just let life happen, right? So you really can't control everything. So you try to do the best you can, and don't worry. Okay, sure. So we start off with carbohydrate metabolism, and we'll first start off with glycolysis. My dear students, in glycolysis, as we all know, it basically takes place in the cytoplasm. Now, this is glycolysis. It starts off with glucose, then it gets converted to glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, which is a big molecule, is broken down to form glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate is acted upon by an enzyme called glyceraldehyde 3-phosphate dehydrogenase. And this is converted to 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate forms 3-phosphoglycerate. 3-phosphoglycerate forms 2-phosphoglycerate. 2-phosphoglycerate forms phosphoenol pyruvate, and phosphoenol pyruvate forms pyruvate. So pyruvate is the end product of glycolysis if there is oxygen. If there is no oxygen, we find that pyruvate is converted to lactate. Pyruvate is converted to lactate with the help of an enzyme called lactate dehydrogenase. Now let's just see the cycle a little more carefully. The first step is irreversible, the third step is irreversible, and the last step is irreversible. The first step, hexokinase and phosphofructokinase utilize 1 ATP each, so it's minus 1 ATP, right? The first step in glycolysis, which produces energy, is glyceraldehyde 3-phosphate dehydrogenase, right? And here, two molecules of NADH are produced, and every molecule of NADH produces 2.5 ATPs. Therefore, two molecules of NADH produces 5 ATPs. Okay. Next, 1,3 bisphosphoglycerate. You have a substrate level phosphorylation called so that is 1,3 bisphosphoglycerate, which produces 2 ATPs here. And then your pyruvate kinase, also a substrate level phosphorylation, which also produces 2 ATPs. So overall, you have 5 plus 2 plus 2, which is 9 ATPs produced and 2 ATPs which are consumed. So in glycolysis, you have a total of 7 ATPs which are formed. Okay, we'll go on. Now we did the first enzyme called exokinase. So usually they ask you about exokinase and glucokinase. So let's just see this. Glucokinase majorly seen in the liver. Exokinase is seen in all tissues. But the most important difference is glucokinase, as the name suggests, it acts only on glucose, whereas hexokinase acts on the various hexoses like glucose, fructose, mannose. Glucokinase comes into play only after eating food, right? It acts when the blood glucose level is high, whereas hexokinase can act even when the blood glucose level is very, 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 very low. So you see, the affinity to the substrate is very high with hexokinase, whereas it is low with glucokinase. And the Km value is low for the hexokinase, is high for your glucokinase. So there's small differences between glucokinase and hexokinase. Now, we've studied that it ends with pyruvate. Once it ends with pyruvate, your body shifts this to Krebs cycle, right? So this pyruvate is then converted to form something called acetyl-CoA with the help of an enzyme called a pyruvate dehydrogenase multi-enzyme complex. And this acetyl-CoA now enters your Krebs cycle. Acetyl-CoA combines with the oxaloacetate to form citrate, hence citric acid cycle. Okay, and then citrate forms cis-aconitate, it forms isocitrate, it forms oxalosuccinate, forms alpha-ketoglutarate, forms succinyl-CoA, succinate, fumarate, L-malate, and oxaloacetate. Back again. Okay, so oxaloacetate is the first product to be used as well as the last product to be formed in Krebs cycle. Okay, next. What are the steps in which energy is produced? So the simple password called alpha miss is a silent teacher. ST is the substrate level phosphorylation, which stands for succinate thiokinase. The first three steps produce a lot of energy. That is, alpha ketoglutarate is, with the help of two molecules of NADH, produces 5 ATPs. M is malate dehydrogenase, produces 5 ATPs. I is isocitrate dehydrogenase, produces 5 ATPs. Succinate dehydrogenase is here, which with the help of FADH, a single molecule of FADH produces 1.5 ATPs. So two molecules of succinate dehydrogenase produces 3 molecules of ATP. Succinate thiokinase produces 2 molecules. This is the only substrate level phosphorylation in citric acid cycle. So that makes it 15 plus 3, 18, 18 plus 2, 20. So that is the total number of ATPs produced in Krebs cycle. 5 ATPs which are produced in this step between pyruvate to acetyl-CoA. So let's recap this and see what are the steps in which energy is produced in both of these pathways, right? In glycolysis, hexokinase and phosphofructokinase is -1 and -1. Glyceraldehyde 3-phosphate dehydrogenase is 5 ATPs. 1,3 bisphosphoglycerate pyruvate kinase, both substrate level phosphorylation, produces 2 plus 2, making it 9 ATPs. Pyruvate dehydrogenase, that is the conversion from pyruvate to acetyl-CoA, produces 5 ATPs. All these are in your Krebs cycle, right? Your alpha miss, that is alpha ketoglutarate, malate dehydrogenase, isocitrate dehydrogenase, succinate dehydrogenase, and succinate thiokinase. I told you succinate thiokinase is the only substrate level phosphorylation in Krebs cycle, whereas in glycolysis you have 2 substrate level phosphorylations. So this makes it so a total of 32 ATPs in your glycolysis. If you see the older pathway, the older calculations, it was 38 ATPs because each molecule of NADH produces around 3 ATPs, right? But they found that it is not 3, it's just around 2.5. Similarly, for your FADH, it was 2 according to the older calculations, but now it is 1.5 as per the recent calculations. Okay, next. Now, if the glycolysis is occurring anaerobically, then we find that you don't have this glyceraldehyde 3-phosphate dehydrogenase at all. This big step is missing here, okay, which produces 5 ATPs. So you have the 2 substrate level phosphorylation, which produces 4 ATPs. 2 ATPs are consumed, so that is a total of 2 ATPs only if it is anaerobic. Okay, right. Next one. Now, citric acid cycle is said to be an amphibolic. That is, there is both synthetic as well as catabolic processes that is happening in your that is both anabolic and catabolic pathways are going on in citric acid cycle. Then there is also another step called anaplerotic, means to say, anaplerotic means, see what happens is in citric acid cycle, for example, oxaloacetate is used up in citric acid cycle. It is used for various other products. For you see, for example, oxaloacetate can get converted to form aspartate. So it is moving away, right? But you need a citric acid cycle to go on. So the citric acid needs these intermediaries. So they are filled up. Like for example, pyruvate is acted upon by pyruvate carboxylase and forms oxaloacetate. So this is an anaplerotic reaction. Pyruvate is acted upon by malic enzyme to form malate. This is an anaplerotic reaction. Glutamate can be converted to alpha ketoglutarate, either with the help of a transamination, what is going on, or with the help of glutamate dehydrogenase. It can form alpha ketoglutarate, right? So this is how, my dear students, your citric acid cycle should be understood here. Right, so you see citrate, how it's going to form fatty acids and sterols, or alpha ketoglutarate forms glutamate, and all the this particular pathways are very, very important. If you see here, glutamate acted upon by transamination to form alpha ketoglutarate. Similarly, oxaloacetate undergoes transamination to form aspartate. It can also be the other way around. Okay, right. Cori cycle. When you're exercising and when you're running, there is less amount of oxygen. If there is less amount of oxygen, what happens is glucose is converted to lactate. right? Now this lactate enters the bloodstream, from the bloodstream it goes into the liver, and this lactate is converted back to form glucose with the help of gluconeogenesis. And so this conversion from lactate to glucose is what is called as gluconeogenesis, right? And this glucose re-enters the bloodstream, goes back into the muscles so that you can keep on running. On the other hand, you have another pathway called glucose-alanine cycle. Now, this is also called Cahill cycle. Now, what happens here is basically your nitrogen from the amino acids, right? Glucogenic amino acids, on the other hand, mostly it is alanine. So, all your pyruvates are then converted to alanine in the muscles. Alanine enters the bloodstream, goes into the liver. Alanine is reconverted to form pyruvate. Pyruvate forms glucose. Glucose comes back into the muscles. So Cahill cycle mostly takes place during the periods of starvation. Cori cycle takes place when you're running. Lactate is the most important ingredient there. And here it is alanine. Cahill cycle, C-A-H-I-N-N. Okay. Okay, now we move on to gluconeogenesis. Gluconeogenesis is the formation of glucose from non-carbohydrate sources, right? Your brain needs just glucose. So body uses whatever is available in and around to convert it and form glucose. Okay, now if you see glycolysis, there are only 3 irreversible steps. So these 3 irreversible steps are overcome and you have glucose over there, right? So now, where is pyruvate? Pyruvate is inside your mitochondria, right? Because pyruvate now has to enter your Krebs cycle, if you remember the glycolysis. Now, in gluconeogenesis, pyruvate has to overcome the last step if it's going ulta, if it's going reverse, right? Pyruvate is converted to form oxaloacetate, if you observe here, with the help of an enzyme called pyruvate carboxylase, which is the first most important enzyme in gluconeogenesis. My dear students, I'm not teaching you. Okay, gluconeogenesis, I take 1 hour to teach. Okay, this is just 5 minutes of recap. Okay, now this oxaloacetate is converted to malate, and this malate now moves out of the mitochondrion. Malate is reconverted to form oxaloacetate with the help of malate dehydrogenase, and now oxaloacetate is converted to phosphoenol pyruvate with the help of an enzyme called phosphoenol pyruvate carboxykinase. That is the second important step in your gluconeogenesis. Once phosphoenol pyruvate is formed, it goes back to 2-phosphoglycerate, 3-phosphoglycerate. All of these are reversible steps. It goes up all the way until it reaches fructose 1,6-bisphosphate. If you remember, phosphofructokinase was an irreversible step, right? Now, this is acted upon by fructose 1,6-bisphosphatase, which is the third most important enzyme in gluconeogenesis. It forms glucose 6-phosphate, which is now acted upon by glucose 6-phosphatase, and it ultimately forms glucose. So this whole process of conversion of pyruvate to glucose, right, is gluconeogenesis from non-carbohydrate sources like glycerol, right, lactate. All of them can form alanine. All of them can form glucose. So this is how basically we try to understand what is going on in gluconeogenesis. And gluconeogenesis, my dear students, majorly takes place in the liver and to a certain extent in the renal cortex. Okay. Now, when you think about gluconeogenesis, we should know a few important things. Now, for example, gluconeogenesis is inhibited by insulin. Insulin majorly inhibits gluconeogenesis. ADP inhibits gluconeogenesis. What are the activators of gluconeogenesis? The activators of gluconeogenesis are cortisol. Then you have acetyl-CoA, right? And then you have adrenaline. And then you have glucagon, right? Glucagon. You have adrenaline. All of these are activators of gluconeogenesis. Most importantly, pyruvate carboxylase, okay? Whereas the inhibitor is an insulin. So please understand this well. Right? Now we have a concept called as GLUT or glucose transporters. GLUT 1, 4 and 5 are most important. Obviously they can ask you anything else. GLUT 1 because that's most commonly seen in the RBC. Of course your brain and all that, but RBC, please remember that. GLUT 4 is most important because insulin increases the amount of GLUT 4. right? And GLUT4 is most commonly seen in the adipose tissue, in your, that is your fat tissue, your skeletal muscles, your heart muscles, all of them is GLUT4. GLUT5 is important for as a fructose transporter, So in the sperms, in the small intestine, in the testis, it is GLUT5 which is more important. Right, GLUT3 is your neurons and brain. GLUT2 was asked for the medical students. The beta cells of pancreas was asked once for the exams. Okay, so you have the various forms of GLUT1, GLUT2, GLUT3, GLUT4, and GLUT5. So GLUT4 being most important. Okay. Now about glycogenesis and glycogenolysis. Now, my dear students, when glucose is collected and tied up together, it forms glycogen, right? So the only difference between glucose and glycogen is lots of molecule of glucose which are branched is glycogen. These linear strands, if you remember the class, are 1,4 bonds, and when you have a branch, it's a 1,6 bond. Okay? Now, how is glycogen formed? Glucose is converted to glucose 6-phosphate. It's converted to glucose 1-phosphate. It's converted to UDP glucose with the help of an enzyme called UDP glucose pyrophosphorylase. Now UDP glucose is ready. It's like a carrier. It goes and attaches itself to a glycogen molecule, a pre-existing glycogen molecule, if it is present, or any protein like glycogen. Okay, so it goes and attaches itself to a pre-existing glycogen molecule, and then comes the most important enzyme called glycogen synthase. Glycogen synthase is very important for the formation of 1,4 bonds, and it increases the length of glucose like this to form glycogen. Okay, and glycogen synthase is activated by insulin. Right now, but this is still not glycogen because you don't have the 1,6 bond, you don't have the branch there. If you want the 1,6 bond, if you want the branch to be formed there, then you need a branching enzyme. Now, the branching enzyme is it breaks the 1,4 bond, makes the 1,6 bond, and that is why the branching enzyme is called glucosyl 4,6 transferase. He is the branching enzyme, right? Well, if you see, these are a few important points which you should know and you should remember about glycogenesis. On the other hand, if you're talking about glycogenolysis, if glycogen is broken down, right? For example, you're fasting, your glycogen is stored in the liver, right, and in the muscle. But per tissue, like if you're seeing per weight, as per weight, if you're seeing maximum amount of glycogen is present in the liver. But if you see overall, simply because muscle is a larger mass than liver, then the maximum glycogen is present in the muscle. But if they ask you as per the weight, per weight, then it is always the liver. Now, if you look into it, my dear students, glycogenolysis is when glycogen is broken down to produce glucose, right? The first step in this is the help of an enzyme called glycogenphosphorylase, right? Now, what you should remember about glycogen is that it's a big structure like this, right? A lot of glucose molecules. Now, glycogen phosphorylase is important because it breaks these 1,4 bonds. It breaks these 1,4 bonds. Glycogen phosphorylase breaks these long 1,4 bonds, and it releases these glucose molecules by converting them to form what is called as glucose 1-phosphate. This forms glucose 6-phosphate, and this forms glucose. Wish glycogenolysis would be this simple, but it is not. Okay. Now, that's the first step. So the most important step for glycogenolysis is glycogen phosphorylase. He does the major cutting part, right? And glycogen phosphorylase is activated by epinephrine and glucagon. Please remember that. Okay. Right. Now, this glycogen phosphorylase can only cut the glycogen until there are 4 glucose molecules adjacent to a 1,6 bond. Now, this structure is what is called as limit dextrin. Okay. Now you have a debranching enzyme. What is the function of this debranching enzyme? The debranching enzyme should break these 1,4 bond and also break this 1,6 bond. And that is how the branch chain is broken. So now the most important debranching enzyme who comes in, right? He breaks this 1,4 bond here, attaches these 3 glucose molecules here. So that is why this debranching enzyme is called as glycosyl 4,4 transferase. It's very easy. It's very, very simple. Okay. Then you have a 1,6 bond, And ultimately, this 1,6 bond is broken by an enzyme called as amylo alpha 1,6 glucosidase. This is the enzyme which comes and breaks this 1,6 bond and liberates this glucose molecule, which is free. Right? So in this process, your glycogen is broken down. Let me summarize. The most important enzyme for glycogenesis is glycogen synthase. On the other hand, the most important enzyme for glycogenolysis is glycogen phosphorylase. Okay, so let's move on. Glycogen storage disorders, mucopolysaccharidosis disorders, all these are very, very important, so you should remember them. I'll just touch upon these. There are 6 major types here. Von Gierke's disease, Pompey's, type 3 is Cory's, type 4 is Anderson's, type 5 is McArdle's, type 6 is Hertz. Right? Von Gierke's disease is due to deficiency of glucose 6 phosphatase. Hepatomegaly is a very important feature. Pompey's disease is lysosomal maltase. Cory's disease is because of a debranching enzyme. Anderson's disease is because of the branching enzyme. Now you understand what is branching and debranching enzymes. McArdle's disease is due to muscle phosphorylase, and Hertz disease is due to liver phosphorylase. Okay, deficiency of these enzymes results in the glycogen storage disorders. Of course, the most important glycogen storage disorder is von Gierke's disease. Next, let's do a few MCQs so that it gives you a better understanding of carbohydrate metabolism. The product of glycolysis in erythrocytes. So, they don't have your mitochondria and all that. So the most important thing here is the product of glycolysis in erythrocytes is your lactate. Next, glucokinase is more active after a meal. I think we have already studied. It is an inducible enzyme, right? Right. Which enzyme catalyzes an irreversible reaction? Remember those 3 irreversible reactions we studied? Exokinase, phosphofructokinase, and pyruvate kinase. An example of substrate level phosphorylation is, right? So you have, this is in your glycolysis, you have 2 of them. There is phosphoglycerate kinase and pyruvate kinase. These both are examples of substrate level phosphorylations in glycolysis. Whereas in your Krebs cycle, it is your succinate thiokinase. Okay. Which enzyme's glycolytic pathway is inhibited by fluoride ions? The answer is enolase. One dental question. Okay. Next. As the end product of glycolysis, pyruvate and NADH are produced. During anaerobiosis, the NAD is regenerated by, if you remember the last step in glycolysis we learned, this pyruvate is converted to lactate with the help of lactate dehydrogenase. How does this do? It takes the NADH from there and is reconverted to NAD. That is what he's asking in this question. So the answer for this is lactate dehydrogenase. ATP is generated in all the steps. We have studied all the steps in which ATP is produced. Hexokinase, ATP is consumed. Okay. Rappaport-Lubbock cycle is a small variant of glycolysis, like we studied Lahey, like Cahill's and Cory's and all that. Rappaport-Lubbock cycle occurs in glycolysis, wherein 1,3 bisphosphoglycerate is converted to 3-phosphoglycerate, right? 3-phosphoglycerate. In this process, 1,3 bisphosphoglycerate is converted to 2,3 bisphosphoglycerate, which is then converted to 3-phosphoglycerate. Now, this 2,3 bisphosphoglycerate is very, very, very important because it reduces the affinity of hemoglobin for oxygen. So when you're exercising, hemoglobin is having all the oxygen, right? So when you're exercising, you don't want the oxygen to be in the hemoglobin. You want it to be in the muscles, right? So when you're exercising, there's an increase of 2,3 BPG. This cuts the oxygen from oxyhemoglobin and makes sure that your the oxygen is then entering your tissues. That is the most important function of Rappaport-Lubbock cycle. Okay, so the answer is 2,3 BPG enhances oxygen dissociation. Okay, next. There is no net synthesis of glucose from fatty acids simply because of the irreversible step pyruvate to acetyl-CoA. It takes place with the help of a pyruvate dehydrogenase multi-enzyme complex. It needs 3 enzymes, mainly pyruvate dehydrogenase, right? And 5 coenzymes. Which one? pyruvate dehydrogenase multi-enzyme complex. What are the coenzymes? NAD, FAD, coenzyme A, TPP, lipoic acid. Done. Okay. Gluconeogenesis is inhibited by, now this we have done it. I've already told you it is inhibited by insulin, but it is activated by cortisol, it's activated by adrenaline, activated by glucagon, right? And all that. All of the following are substrates of gluconeogenesis except lactic acid, alanine, glycerol, all of them are not palmitic acid. Which glycolytic enzyme uses inorganic phosphate? Remember that big enzyme which produces maximum energy in glycolysis? Your glyceraldehyde 3-phosphate dehydrogenase. He's the one who needs an inorganic phosphate ion. Okay, so the answer is glyceraldehyde 3-phosphate dehydrogenase. Gluconeogenesis, as we all know, is liver or a little bit in the renal cortex. So, liver. Which enzyme generates? Here he is talking about your HMP pathway or hexose monophosphate shunt pathway. Okay. Now what happens here is you have to know a little bit about HMP pathway. HMP pathway begins with glucose 6-phosphate. It's divided into an oxidative and a non-oxidative. Oxidative means two molecules of NADPH are produced. It's not NADH, it's NADPH, and you have a pentosis which is being one pentose, okay? So two molecules of NADPH, one pentose being produced. This is the oxidative pathway. The non-oxidative is nothing but it produces like trans-ketolase, trans-aldolase. These are the most important steps in non-oxidative pathway, which produces a lot of intermediaries which are basically formed. Okay, so in oxidative you have NADPH and pentosis. Why do you need NADPH? Please note, in HMP pathway, ATP is not consumed, ATP is not produced, right? HMP pathway, the major function of hexose monophosphate shunt pathway or pentose phosphate pathway or Harburg-Dickens pathway is to produce NADPH and is to produce pentosis, right? So let's just run through why your body needs NADPH. Now, NADPH is required for the formation of fatty acids, cholesterol, steroid hormones, free radical scavenging, maintaining the RBC membrane integrity. Very important. Let's look at your RBC membrane. It produces, there's a lot of reactive oxygen species which are produced. What do these do? They remove the electrons from the cell wall. Now, you don't want that to happen. You want these reactive oxygen species to be normal, right? You don't want them to be hyperactive, right? So you need something called as glutathione dehydrogenase, right? It's a very important what you call as antioxidant enzyme, glutathione dehydrogenase. Now, if you want glutathione dehydrogenase to be active, you need it to be in a reduced form. You need NADPH. NADPH makes sure your glutathione dehydrogenase remains in a reduced state, right? So if you don't have NADPH, your glutathione dehydrogenase is your antioxidant enzymes are not functioning. Therefore, the reactive oxygen species becomes hyperactive. They act on the cell wall, the RBC breaks, and it results in what is called as hemolytic anemia. This is how, my dear students, if you look at the HMP pathway, the most important step in HMP pathway is your glucose 6-phosphate dehydrogenase. This is the first step which comes in, not shown here, but the first step in HMP pathway is G6PD. So, G6PD deficient individuals, as we say, is because of the HMP pathway. So, if they have a G6PD deficiency, they land up with, they can land up with hemolytic anemia. And if you're taking drugs like Primaquine, I think we have an MCQ there. If you're taking drugs like Primaquine, it increases the amount of reactive oxygen species, right? So, and you don't have G6PD. Your body does not have NADPH. Glutathione dehydrogenase cannot be in a reduced state. RBCs break down. It results in hemolytic anemia. Okay. So there's a few things like detoxification by hydroxylation, maintaining the transparency of the lens, bactericidal activity of macrophages and all that, right? And pentosis, you have ribose 5-phosphate. These are pentoses which are produced. So NADPH, the HMP pathway is majorly known for NADPH and pentosis. So this we have done. Deficiency of G6PD would cause hemolytic anemia. I think I've explained. The hormone activating phosphorylase, your glycogen phosphorylase, is epinephrine. We have learned those, right? What is glycogen? Having unbranched or straight line? No, it's a branch structure, right? The best answer is that it is seen in the brain. Okay, it's majorly seen in astrocytes. Okay. Next, which enzyme is active in the phosphorylated state? Now, this is your glycogen phosphorylase. Glycogen phosphorylase is active. Glycogen phosphorylase, your enzyme for glycogenolysis, is active in the phosphorylated state, but it is normally present in your unphosphorylated state. When it gets phosphorylated, that is when your glycogen phosphorylase becomes active. NADPH is required for all. So we've given a huge list where all NADPH is needed, except substrate for electron transport chain. But respiratory burst, hydroxylation, detoxification of reactive oxygen species, it's used in all these areas. Which coenzyme is used for the HMP pathway? Very important is TPP, thiamine pyrophosphate, for transketolase reactions. Okay. Next, which of the following can precipitate a G6PD deficiency? As I've said, it is primaquine. Most important is primaquine, antimalarial drug. What cannot be converted back to glucose? The answer is acetyl-CoA, because of that particular step, pyruvate to acetyl-CoA. What is the purpose of uronic acid? These are small pathways which are present. Why do you need a glucuronic acid basically? Why do you need it? Simply because it helps in the conjugation of bilirubin, steroids, and synthesis of GAGs or glycosaminoglycans. Okay, it's used for the conjugation of bilirubin, steroids, and GAGs. What hormone controls the HMP pathway? The most important hormone is insulin. Okay, insulin stimulates the pathway by activating the key enzyme, right? So please remember insulin. Next, what is essential pentosuria? Now, essential pentosuria is the excretion of L-xylulose or a pentose in the urine. That's why it's called pentosuria. And why do you have it? Why do you have it? It is because of a deficiency of an enzyme called xylulose reductase, or it's an enzyme called xylitol dehydrogenase. Xylitol dehydrogenase. Okay, these are two enzymes, xylitol dehydrogenase and xylulose reductase, which are important for essential pentosuria, because there is an excretion of L-xylulose, right? Okay, so this was just in brief, very, very small. We went through glycolysis, HMP pathway, glycogenesis, glycogenolysis, HMP pathway, gluconeogenesis. We've done all of this. I told you the most important points. So I'm hoping that I've done some justice in these 15 minutes. Otherwise, this is a big 3 to 4 hours lecture, so you understand each and every step. This is just to give you a small preview just before your exams, if you just want to go through, and I think it will help you. Okay, all of you, thank you so much. Take care. God bless.